Okay, so this is chapter 5, part 3, and our focus here is going to be on active transport. This is going to be transport that requires energy. We'll talk about bulk transport as well. Okay, so active transport, as I just said, requires energy, and that's going to be in the form of ATP, adenosine triphosphate. We've seen that molecule before now. Inactive transport substances, whatever that substance is going to be moved against their concentration gradient. We call active transport proteins, we call them pumps. And that's because they're literally pumping whatever it is, an ion or a molecule, against their concentration gradient across that membrane. Active transport pumps are required by cells. Their function is to maintain concentrations of ions and other substances in the face of passive transport and diffusion. Okay, so we have molecules that are naturally passing through the membrane, either straight through the membrane or through a carrier or a channel protein, via passive transport or facilitated transport from high to low. And so these pumps have to keep going in order to maintain concentrations to fight against passive transport. And so the textbook example of an active transporter is called the sodium-potassium pump. And we're going to see this, again, this is the textbook example. It's going to be our example of an active transporter. It's also going to be involved in establishing something called the electrochemical gradient, which is actually on the next slide. So let's take a peek here. So we've got our phospholipid bilayer, right, our plasma membrane. We've got outside the cell, and we've got inside the cell. So here's the cytoplasm. And then we have this membrane transporter, okay? We have sodium, Na+ ions, that's these, okay? And we also have potassium ions, K+, and that's going to be represented by these yellowish ovals, okay? And if we look at concentration, so we have sodium ions, a high concentration of sodium ions outside the cell, and a lower concentration of sodium ions inside the cell. And then if we look at potassium, it's the opposite. So we've got a higher amount of potassium ions inside the cell and a lower amount of potassium outside the cell. Okay, so think about that. Think about those concentrations and natural tendencies of diffusion or passive transport. So our active transporter is going to pump against those natural gradients, okay? And so here we go. So here we go. This is the first conformation or the first shape here. And so we can see that the hydrophilic core of this is going to allow 3 sodiums, so 3 Na+, to bind to the inside of here, okay? That binding, along with the help of ATP, so energy, is going to cause that protein to change shape and to dump off those sodium ions on the outside of the cell, so on this side of the membrane. So notice that sodium was just pumped against the concentration gradient. Sodium was pumped from low to high concentration against the gradient. Energy was needed for that, so there's that ATP. On this side of the membrane now, potassium ions, 2 of them, will bind again to the hydrophilic core of this carrier protein. Okay. ATP works here as well, causes this to change shape or conformation, and those potassium ions are dumped off on the inside of the cell. Okay. What also happens? Well, so ATP, it gets cleaved, and so we end up with ADP, adenosine diphosphate, and what we call inorganic phosphate. Okay, so we're going to learn a little bit later on kind of how ATP works in chapter, I think, 7. But again, ATP is required for this process, and what you're doing is you're building a concentration gradient. You're going against the concentration gradient. Okay, so again, 3 sodiums are pumped out and 2 potassiums are pumped in. And again, if we look at concentration, again, we're building the sodium concentration outside of the cell, and we're building the potassium concentration inside the cell. Okay, so again, that's our textbook example of an active transporter or a pump, an ion pump. And again, it's going to pump 3 sodiums out and 2 potassiums in. And that's going to help to set up this, which is called the electrochemical gradient. And so when you hear that term, this is going to be the combined effects of both concentration gradients and electrical gradients. So you have to think about that. When you're moving ions, you're moving charge. And so you end up with a charge difference, okay, an electrical difference across the membrane. And in living cells, it's very common that there's going to be a charge difference across the plasma membrane, and it's really important to the functioning of many cells in our bodies. Because of the sodium-potassium pump, really that's the major player that's setting up this electrochemical gradient. So remember, 2 sodiums get pumped out for every 3 positively charged sodiums get pumped out for every 2 positive potassiums that get pumped in. And so therefore you're putting out 3 and you're only bringing in 2. And so what you end up with is the exterior of the cell, so the extracellular fluid here, overall, it's going to have an overall positive charge. And if we look at the inside of our cell, our cytoplasm here, overall, it will have a net negative charge. And again, this is mainly established by the sodium-potassium pump. There are, more sodium positively charged ions being pumped out than positively charged ions being brought in. Okay. Now, on both sides of the membrane, there are positive and negatively charged ions. It's just the distribution is unequal. So there's more positive outside and less positive inside. Okay, so it gets an overall negative charge. Okay. So when we talk about active transport, again, we're talking about the use of membrane proteins, right, that are going to move substances from low to high concentration. In this case, all of the membrane proteins involved are carrier proteins. There are no channel proteins that will do active transport. Okay, and materials cannot be actively transported across the membrane without a protein. And so proteins are required for this. So again, we have carrier proteins only. The sodium-potassium pump was a carrier protein, right? It changed shape as it brought materials into or out of the cell. There are 3 types of carrier proteins involved in active transport. There's uniporter, symporter, and antiporter membrane proteins. So a uniporter is going to carry one specific molecule or one specific ion. A symporter is going to carry 2 different molecules or 2 different ions. But importantly, a symporter is going to carry those in the same direction, either into or out of the cell. The third type, the antiporter, again, carries 2 different ions or 2 different molecules, but it's going to carry them in opposite directions. And so the sodium-potassium pump that we just looked at a few slides ago is an example of an antiporter. A carrier protein changes shape, does active transport, carries sodium in one direction, potassium in the other direction. So it's an antiporter. Okay, so what we've talked about so far is primary active transport. Okay, and I'll show you this again on the next slide. So in primary active transport, we're moving substances, ions or particles, across the membrane, right, from low to high against the concentration gradient. And primary active transport sets up a gradient and it sets up a charge difference across the membrane. Primary active transport, remember again, uses ATP directly. So I've already showed you this, right? The sodium-potassium pump is an example of primary active transport. There is another type of active transport that results from the electrochemical gradient established by primary transport, and that's called secondary active transport. And so again, I'm going to show you an example of two examples of this. So in secondary active transport, we're still moving materials against their concentration gradient, but instead of using ATP directly, we use the electrochemical gradient that was established by primary transport. Okay, so again, it's moving molecules against their concentration gradient. That really should be written here. You might want to add that to your notes. So it's the movement of materials, ions, particles, whatever it is, against their concentration gradient. That requires energy, but that energy is not going to come from ATP. It's going to come from the electrochemical gradient set up by primary transport. Okay, so let's look at both of these again. So this is just a different picture, again, showing the sodium-potassium pump. So it's the same transporter that I showed you a few slides ago. This is actually from our textbook that we're using as a reference. And so I just wanted to include this picture. So it goes from A, B, C, D, E, F, kind of in a loop. So again, 3 sodium ions will be picked up from the inside of the cell where the concentration of sodium is low. They will bind to this carrier protein. ATP jumps on, causes the protein to eventually change shape. And when it changes shape, it will then dump out those sodium ions on the outside of the cell. So those are 3 positively charged ions being brought out of the cell. It will then pick up 2 potassium ions, 2 positively charged ions, and will change shape and dump those off inside the cell. Okay, so again, ATP was used. We pumped against the concentration gradient. This again is an example of an antiporter. And what we're building here is not only a concentration gradient, but also a gradient of ions. And so again, we call this an electrochemical gradient. Because 2 pluses go out and only 2 pluses come back in, remember the outside of that membrane is going to have an overall positive charge, while the inside of that membrane will have an overall negative charge. And so again, this is the main pump involved in setting up the electrochemical gradient. So we call it an electrogenic pump. Okay, so there's our example of primary active transport. Let's look at secondary active transport. So again, we're still pumping against a concentration gradient instead of down it. Okay, and it's going to require energy, but instead of energy in the form of ATP, we're going to use the established electrochemical gradient. And so in this figure, here's that sodium-potassium pump again, doing its job, right? Taking sodium out and bringing potassium in. I'm showing it doing both at the same time here, but remember it's one at a time. So this is an antiporter, setting up an electrochemical gradient. Here's the secondary transporter. So that's over here on the right. This is called the sodium glucose symporter. So symporter, it's bringing in 2 different molecules in the same direction. So in this case, bringing them into the cell. So it's moving 2 different molecules in the same direction. And so here's glucose. Okay, this blue hexagon. And you can see that there's a higher concentration of glucose inside the cell. And so we've got to pump it. The only way to get glucose from the outside of the cell in is through active transport. Okay, and in this case, it's going to be secondary active transport. So the sodium glucose transporter just needs sodium to move. And so because there's already this gradient of sodium, sodium is going to naturally go from high to low. And so when sodium binds and glucose binds, this thing will dump both of those on the other side of the membrane. And so this secondary active transporter is using the sodium gradient to bring in glucose. So again, it's secondary active transport. It's the gradient of sodium that's the source of energy here. It's the source of what we call potential energy. Okay. Here's another example, same idea. We got primary followed by secondary active transport. Okay, so let's look at this. So we've got a proton pump. So this protein in the membrane is going to pump H pluses, protons, right, from the inside of the cell to the outside of the cell against the concentration gradient. That requires ATP. So this is our example of primary active transport. When this thing does its job, it sets up a gradient. It establishes a gradient. So there's going to end up being a high amount of protons outside the cell compared to the cytoplasm of the cell. So down here in the membrane, there is the sucrose proton cotransporter, another example of a symporter. And so the power, the energy source for this protein to work comes from the proton gradient. And so protons will have a natural tendency to go into the cell. And the only way that they're getting in through this protein is if sucrose comes with it. And so both the proton as well as the sucrose molecule will bind, and both will be transported across the membrane. The protons are moving down their concentration gradient, while the glucose is being pumped against its concentration gradient. Okay, so again, that is our second example of secondary active transport. And that's it for active transport.